Thank you for being back. So, after the rocky seashore picture show and metazoan biology and macroalgae, we will now dive into the plankton world. And I'm happy to introduce and leave the stage to Daniel Vaulot, who will take us on a trip through plankton.
C'est bon? Bonjour à tous. Merci Myriam pour l'introduction. Deux choses. Je vais, comme Patrick et Philippe, je ne vais pas remercier tout le monde. Enfin, je ne vais pas citer tout le monde, ni remercier tout le monde. Hein? Ouais, ah oui, in English. I'm going to switch to English. Sorry. Ah ouais, j'étais parti en français, je ne sais pas pourquoi, parce que ma dernière conversation était en français en fait, avec une Française. Donc, I'm going to switch to English, and so as the previous speakers, I'm not going to be acknowledging everyone nor thanking everyone, but I'm going to try to have a rather personal history of plankton in Roscoff. Just to thank all the organizers, and especially Catherine and everyone. This is super nice. I mean, this is very, very well organized. It's very super nice. So this started really bad because Gérould in Science in 1899, in Science, huh? It was going around the biological station in France and especially in Brittany, and he noticed that the hygienic advantage of the ocean voyage, I think that's all the things that you smell from the algae and so forth. He didn't mention the dark side also of Brittany, but he said, as regard to fauna, the fact is to be emphasized that for plankton studies, Roscoff is badly situated, whereas shore collecting for macroalgae, it's very good. So no macroalgae in Roscoff. And this was pointed to me by John Dolan. John Dolan is from Villefranche, and he's doing an amazing work on the history of plankton. He's doing really amazing work. So he found this. But it was not the only person, because Georges Tessier in 1962, Drash had sent 5 students to Roscoff to study plankton. And when they arrived, they had a dismal arrival with Tessier, who told them, vous venez étudier le plancton de la Manche, mais il n'y en a pas. And this is the Guy Jacques, one of the students, who is in his memory. He said, so you came to study plankton, but there is none. We thought he was meaning little, but... And there was absolutely nothing to discover for plankton there. So this is bad. So I'm going to stop here. So no plankton and no need to study plankton. But actually, the plankton researchers in Roscoff have now published more than 800 papers, almost 800, but I think it's more now, including in Science, in Cell, in Nature, ISME, eLife, PNIs, and so forth. And this is, as you see, some kind of increasing trend. So I think if we celebrate the 200th anniversary, we'll probably have a bit more knowledge of plankton. Not always in Roscoff, but at least it's a very good place to study plankton. What are the pioneers? The pioneers is although he was working on sand dinoflagellates, but Balech came to Roscoff and began to study with his microscope some of these planktonic organisms, and actually he named one species, Roscoffia capitata. So that's, for me, it's one of the first to study plankton, although he was more studying plankton from the sand here, or plankton, no, algae, microalgae from the sand. But the pioneers is really this collective thesis. At that time, you could do a collective thesis. This is unknown now. So 5 people were sent like a commando to Roscoff to study plankton. They were Jean-René Garal, who unfortunately deceased very early, and Guy Jacques. And then Francine Baudot, Claude Razoul, and I don't know what the first name of Thierry is. So 2 for phytoplankton and 3 for zooplankton. So this time they thought that zooplankton was more diverse than. So this is in the 100-year anniversary book. This is what Drash was saying. They were going there to study plankton. So, and I'm going now to focus for a bit on phytoplankton, because that's what really developed where we have been our forte. So, sorry. They were the first to describe the bloom of phytoplankton of Roscoff, and especially that there are 2 waves of phytoplankton bloom, one in the spring and the other one in the autumn. And they focus on diatoms, because a bit dinoflagellate, but mostly diatoms, because they were using either method with a microscope, and this was what was available at the time. But they describe actually things that we now are been continuing describing using more modern method. So the plankton group, what we call the plankton group, who changed many times name, now it's called EcoMap for Ecology of Marine Plankton, was really starting in the 80s with Serge Poulet, who was a zooplankton researcher. You can see him here. A very interesting character, actually. And who he had always this mustache, which was really very impressive to me. It was always very well created. Never could get mustache like this. And he worked on zooplankton, amino acids, and also the interaction with colleagues from Naples between zooplankton and phytoplankton, and they published a paper in Nature. And they also collaborated a lot with Plymouth. I'm not going to mention it too much, but all this work I will be mentioning are work in collaboration. I mean, Roscoff, it's nice, but if you only stay in Roscoff, I mean, you are not going to go anywhere. You need really to collaborate, and all these were done with other stations, all the work. Then came Alain Saugnac, who was mentioned yesterday. Unfortunately, he passed away a few years ago. And he was initially a specialist of dinoflagellates. He had been worked in Madagascar, especially with what was is now the ARD. And he came to Roscoff in '84, and he studies the fronts, but more importantly, he's the one that established the Roscoff time series, actually. He was also a philosopher. He published several books that you can find on Amazon. I tried to open one, and I could never pass the first page, but it's not my stuff, philosophy, but he was a real philosopher, and he was a very good taxonomist as well. And then I arrived, 84, and yeah, no bird, but long hair. And what you see here, this piece here, is the first PC ever in Roscoff. Because before there was a Centre d'Informatique, but it was like in a room, which is, and you had to go there, and Pierre Lasserre was very nice to offer me a PC. I had to go to his office and plead a bit, but then we got the first PC ever, IBM PC. So since then I never switched back to Mac. I mean, and you can see the diskette. We had all our data there. So let me go a bit of science, how we get started on phytoplankton. So they told us that there's no phytoplankton of Roscoff. So we say, okay, we are going to go somewhere else. We got position here, so we are going to go somewhere else. And at that time, central oceans were seen as deserts. There was a bit of chlorophyll, but people were thinking that it was dead chlorophyll. And people were thinking that there was nothing in this area. However, in 79, I did my PhD in 80, from 80 to 85. So in 79, just before, John Waterbury, by efflorescence microscopy, discovered Synechococcus. And it turns out that it was a very, very abundant cyanobacterium, and that it was almost everywhere in the ocean. And now, I mean, the group here is really specialized on studying this small bug. At the same, a bit later, using another technique called flow cytometry, Penny Chisholm discovered Prochlorococcus, which turns out to be the most abundant or photosynthetic organism in the ocean. It's abundant. It's typically 100,000 cells per ml in oligotrophic areas. And I happened to do my thesis with Penny Chisholm. Not on Prochlorococcus, because it was before she was doing Prochlorococcus, but I was involved into flow cytometry development, and especially the use of flow cytometry to measure picoplankton. So that's where we began actually to hunt picoplankton using flow cytometry. And again, it was Pierre Lasserre who pushed NRS to invest in this big machine at that time. Now you have seen probably the modern flow cytometers, but this was, a machine that was delivering 1 watt of light. A laser, now you use 20 milliwatt. So this was these big things here, and with Claude Courtis, who was an engineer, previously working with Pierre, we took it to sea during the Sklomas cruise in '87, and there we began to work with cyanobacteria, and especially Prochlorococcus. This is a compte rendu de l'Académie des Sciences on this. We did other cruises, and here you can see a young Fred Partensky in the back. So we had our flow cytometer, it was on a container, and it was on the Discovery in the Med Sea, and we began to study more intensively, actually, these, which were by now called prochlorophyte. At the same time, in 1992, we had a chance to attract here Dominique Marie, who was previously working in Gif Sorivet in a lab working with plants. And he had Breton roots, and he came here, and he really was the one that helped us to develop flow cytometry, especially by developing new methods for example, bacteria or viruses. And these methods now are really standards. So this has a very, at one point, so I mentioned 2 persons, Fred Partensky and Dominique Marie, and they were competing for a number of citations because Dominique, who was just ingénieur d'études at this moment, had more citations than anyone actually in the team. But he just retired, and I think he was really someone really amazing at this with this technique, and he had a huge contribution. I think that was Philippe that was mentioning, and Patrick also, the importance of engineers. For me, that's one of the things fantastic with France, is that you have this support that you don't find in many other countries actually. So, a lot of picture of Fred, huh? So, he's my office neighbor, so I don't want to be after a problem with him. So, what we were studying phytoplankton in the ocean, but we saw very early that it was super important to get things in cultures. And that's when we get Prochlorococcus in culture. And again, Fred had a green hand with Prochlorococcus, because after he stopped trying to isolate Prochlorococcus, we never isolated any other Prochlorococcus, but So we were able to organize workshop where we were combining, especially with our colleagues from Villefranche. Here you see André Morel, who is one of the really pioneer in optical oceanography, and who is, his work has been used a lot now for remote sensing and interpretation of data. So we were able to do a lot of physiological experience with this culture. We also got eukaryotes, and I will come back to this, in culture, and describing new species and even new class of algae, which I've been describing several actually and this is Lorguillou. Not working with picoplankton there, she is in the no-till, going to try to get something from the hydrothermal vents actually. One thing I'm most proud, I don't know if it's to say this, but is that the plankton group has been describing many new species of plankton. And I think that's probably one of the only things that will stay in 50 years, because species stay forever actually. So when you describe a species, even if it was 200 years ago, it's still there. No? I'm not saying that, no, species evolve. Miriam. No, species evolve, but the name, you have to cite the name of the person who describes the species, right? Okay? So these names, some of them are we describe many new species: Bolidomonas, Picophagus, Florenciella, Partonskiella, Lotarella, Chloropicon, Chloroparvula, Prasinoderma, Montoniella, Macromonas, Pseudoaptolina, from many different groups actually. And this is, I think, very critical. Molecular biology and genomics is very important, but describing species is very, very important as well. And this led actually to the Roscoff culture collection, who was founded in 1998. So I'm not sure it's the largest culture collection in the world, but it's one of the biggest for marine algae at least. And now this is Jan, who is in charge of this, with Florence has been contributing a lot, and also now Priscilla. All engineers, either from the university or from CNRS. And this is a resource that we try to make open. And okay, so then came you heard about marine genomics, and the era of molecule. I mean, we had to go molecular. And for us, in the plankton group, this was introduced by two people, actually. Dave Scanlan, who is a very, very good colleague from the University of Warwick, who came at the initiative again of Pierre Lasserre to give a course on molecular approach to microbial ecology. And that's where all of us run our first gels. Actually, we were in that lab that Janine mentioned, which is above the cemetery. And at the same time we were extracting DNA and doing this, they were extracting corpses from the cemetery to move it. So one student fainted, actually. One English student, do you remember, Fred? Yeah, she completely fainted when she saw this. So that was our first experience in molecular biology. And then Wolfgang, who was Hess, who was a postdoc for a very short amount of time, because then he was offered a position at the University of Berlin. So we got molecular. And this led to work, for example, by Fred and now Laurence on genomes. So as we're mentioning before, there's been a lot of genomes, especially for cyanobacteria, but also for eukaryotes. As I mentioned, so I focus mostly on cyanobacteria, but at the same time, picoplankton also was a lot of eukaryotic cells were seen, for example, I'm blessed in myself, by electron microscopy, for which we had no idea. So that's where the molecular, no idea what they were. So that's where the molecular approach arrived. And we, using cloning of the 18th gene, and we discovered that there was a huge diversity of uncultured organism. And this paper, very interestingly, came back to back with a paper from Puri, where she had found independently exactly the same conclusion. So these 5 minutes only? Okay. Oh, okay, I'm going to accelerate. So also, we developed several techniques to visualize these cells using fluorescence, so it's called fluorescing in situ hybridization, developed by Nathalie, and then that was a young Fabrice thesis, where we showed that it was some organism like macromonas were super important in the around here. Actually, that's when we came back to the plankton of the channel. Then when were these tools, and but now there are many more. So beyond phytoplankton, so as a group increase, we got into other groups than phytoplankton only. So for example, viruses, with the arrival of Anne-Claire, where she looked, for example, at the interaction between viruses and diatoms, and Christian Janton, who developed thematics about bacteria, marine bacteria. We had also extended from phytoplankton to protists. So now we are thinking in terms of protists, because it turns out phytoplankton is evolving in many different lineage of the eukaryotic tree, with the arrival of Colomban and with the recruitment of Fabrice. Initially, his office was a bit small. I think now he got promoted since he got director of the UMR. I think he got another chair to be able to get people sitting. And also developing methods about, interaction about symbiosis, parasitism, with work of Laure and Claire about, Aurélie, sorry, Laure and Aurélie, and this is Dominique also. He is also a seller. Not ICs, but so looking at this Amobophrya type. Trends. So what are the trends in the future, what I see? First is going global. I think we changed from, we had this cruise, but I think a big change was really going global, especially with Tara, which provided a wealth of new data and going meta-omics, so metagenomics, meta-proteomics, meta-barcoding, which led to really a whole new knowledge about the plankton, but not locally, but how to define concept and trends across the globe, actually. Using, for example, this was done, so this is a work of Colomban from Tara, and this is a work of the group of Fred and Lawrence on cyanobacteria. One thing that was always important for me and for us, I think, in the group, is to be open, to not to keep our data for us, but to be open. So this was the idea behind the collection, of course, but this was the idea also developing a database, open database, to make everything as open as possible. So, for example, this is a PR2, which I think now is over 900 or 800 citation, which is a database for 18S sequence. PhytoRef, developed by Johan and Colomban here. Johan is here and you will hear, and a database I've been developing recently. And the same thing for genomics with the work of Lawrence and Fred on cyanorac. Also, as I said, it's important to go back to culture. So some of the work, for example, of Christophe has been very important to understand at the cultural level some of the strands that we see in the ocean. Okay, so just to see that this is a work of a group, I wanted to do a collage of all the people that have been in the group, but I said, okay, it's going to be too much. So this was in 2002, 2007, and now we cannot even do a group photo. We need to do some kind, because not everyone is at the same place at the same time. And just for sponsoring, I really want first to thank CNRS and Sorbonne University, but especially CNRS, which has given us so much liberty to do what we want to do, to be creative without any boundary. And having been now in a university in Singapore where things are very different, I bless every day that I've been recruited by CNRS. I was recruited without a thesis. Can you believe this? I was just because I was a polytechnician, so they say, okay, you can go to CNRS. Not anymore. And but EU has been extremely also supportive, and but not only in terms of money, in terms of collaboration, because they force us to collaborate, and this was a blessing, really, to get all this collaboration, both in France and with our sister station like Banyuls and Villefranche, but also with all these other groups like in Naples, in Oslo, in Netherlands, and so forth. So I'll stop here, and if you have any question.
Thank you, Daniel. Thank you, and there's time for a question or two. Anybody?
No, that's good.
I will ask a question. At one stage, you mentioned going molecular. So what is the molecular aspect that you think will bring another step further on these analysis of the taxonomy and biodiversity?
I'm very bad to do a prediction, actually, on what will be the next step of science, because I think it's how do you... François is here. He correct me also with serendipity. Yeah, whatever. Yeah, that you suddenly cross the path of someone or some technique, and then that's like flow cytometry is typically. This was something used in the medical field, and Penichism and a couple of people in the US say, this can be used for plankton as well. And this, like it was probably a discussion, a 2-minute discussion, and then that came. So I never make prediction.
Okay. Thank you. Yes, there's one question over there, even two.
Colombo?
Well, Jan-Hendrik first, and then Colombo.
Jan-Hendrik?
So this is a little bit controversial, but because among friends one can be controversial, I think, sometimes. No, I think this is like it is. Like if you're in a family or with friends, you can also sometimes be controversial. So do you think it is inspiring? Do you think a student who has 24 years and wants to make a really high impact publication in 3 years and wants to contribute something big, looks at a thorough paper with a lot of data and think they can achieve this?
So what's the question?
He the question, as far as I understood, is having these data, these big data, is this students that in 3 years they will be able to reach such big impact paper? That's
well, but I think the thing with high impact paper, and I think the paper by Miriam and Christophe is a very good example of this. You have some high impact paper now where you have 1,000 of authors, and you don't know what the contribution of any of them is actually most of the time. So I think this is a trend and it's not going to change. But I think on the other end, you can also, in a 3-year, if you have a very good idea that nobody had before, you can do a very high impact paper. And you don't have to be for this in a very big group. You have to be in a group where people I was talking to Christophe and said, yeah, we have been thinking about this for 20 years, but we didn't have a PhD. So you could have this science paper 20 years ago, and you will have maybe had more funding for this or not, or you don't care, actually, because you are passionate about what you are doing. So yeah, no, so I think there are the two ways of, yeah.
Yeah, sorry. Daniel, how come you came to Roscoff? What was the trigger?
Well, there was no trigger, actually. I was recruited, I was recruited to work on fish in Montpellier, okay? But I was doing my thesis, so my project at Synechococcus had nothing to do with what I will be doing for the next 35 years. And so I was in the US, and my wife had a position at Ifremer. So I say, well, where I go, I don't want to go to Brest, so what's the next to Roscoff? So that's the way I came to Roscoff. It's just, yeah, exactly. I mean, I could have been, after I said why I didn't choose to go to Villefranche, because, I mean, I like Roscoff, but I would have been super happy in Villefranche as well, I think. The rent is more expensive, but at that time it was okay. I think now to be in Villefranche is better to be in Roscoff for renting.
Thank you very much, Daniel. So